Very good afternoon, everyone. A very hearty welcome to all the speakers and the attendees for this webinar session. On the behalf of Voice of Healthcare and Cipla Diagnostics, I welcome the guest speakers and attendees for this webinar. This episode is a part of our current virtual series of discussion that is with the theme of amplification, reimagining diagnostics with experts in collaboration with Cipla Diagnostics. The topic of this session is chances of emergence of a vaccine-resistant transmissive variant COVID-19. Now, I have Dr. Rohit here from the Medical Affairs team at Cipla Diagnostics. He shall now take this forward from here and start with the introductions. Over to you, Dr. Rohit.
Yeah, thank you, Pinky. So, a very good afternoon to all. For this meeting, we have esteemed faculty with us. Today's session will be moderated by Dr. Biswajit Mohanty, who is a Managing Director at Genex Diagnostics, Bhubaneswar, Odisha. Along with him, we have Dr. V.L. Jayasimha, who is a professor from Department of Microbiology from SS Institute of Medical Sciences and Research Center, Karnataka. Along with him, we have Dr. Kiran Chawla, who is a professor, Department of Microbiology, Kasturba Medical College from Manipal, Karnataka. I welcome all the faculty members for the meeting and would like to hand over session to Dr. Biswajit Mohanty. Over to you, sir.
Good afternoon, everybody, and namaste. Thank you, Dr. Rohit and your young spirited team that you have created such a wonderful platform where many faculties, eminent faculties like Dr. Jaisiman and Dr. Professor Kiran, they will be speaking. They are subject experts. But before that, I'll just speak a few words about this today's topic. Now, to start with a little bit about history, on 30th of January 2020, the first case reported from Kerala, 3 medical students, they returned from Wuhan. On 22nd February 2020, Prime Minister announced one-day nationwide lockdown. On 11th March 2020, WHO declared novel coronavirus disease outbreak pandemic. And 15th March, India declared pandemic. And 25th March, there was a lockdown across India. So this is a little bit about the history. Now, in those days, in those early days, it was a very, very difficult time. Only very, very few labs across India, they were diagnosing the SARS-CoV virus. We have overcome many hurdles like creating diagnostic facilities across India, making the price affordable to common man. Initially, it was started with 4,500, and now in many states, it is coming down to 400. And in government setup, it is almost free. Nobody imagined that in short span that our people can do such a extensive investigations for a population in India, say 1.4 billion people. And then we could manage to make affordable treatment affordable, creating specialized COVID centers, creating treatment protocol, oxygen availability, vaccine development, and successful vaccination program across India. We have done pretty well. Now the state government and the central government has done their work very well. And they played a very crucial role. It is definitely a collective work. New variants are coming up with possible emergence of vaccine-resistant COVID infections. Now today's update, the new case as on today, that is the last 24 hours, 11,466. Active COVID load, 136,683. The death in last 24 hours is 460. And the active coronavirus across India is 0.41%. That is the lowest since March 2021. Perhaps, I don't know whether we are moving from pandemic to epidemic. Well, that's a $1 million question. That's not the thing that we are going to discuss today. Our topic of discussions, well, to start with Dr. Jaisima, we will have 2 eminent microbiologists, and each of them will be getting 15 minutes time to introduce their part. And then after that, we'll have a questionnaire. So each of them, so sir, please stick on to your time because we have a limited time hours available to us. So to start with Professor Jaisima, so he will be talking about the problem statement, various terminology associated with SARS-2 viruses, the immunological responses, variants in epidemiological point of view, and vaccinations in general. Sir, to you. Professor Jaiswal.
Last time we what is it? Am I audible?
Yeah, very much.
Good afternoon, all. Am I audible, sir?
Yeah,
Madam?
Please go ahead.
So good afternoon, everybody, and thanks for giving me an opportunity by the eminent Vishwajit sir and very esteemed Dr. Kiran Chawla, madam, as a co-speaker to me. I think that itself is a wonderful occasion to speak with you or share some important things with you. In fact, I have prepared a lot of slides, but after the time given by the Vishwajit sir, I think I will make it very fast, and I will try to be very crisp, something like that. So SARS-CoV variants, mutations, its importance. I thought I have to put my terminology or the introduction in slides something like this, because a lot of things happened with the SARS-CoV virus. Excuse me. Change the slides. Okay, so my main objectives will be the virus and its structures, mutations, and the variants, what I will be talking about. So the pathogen or the problem statement of the SARS-CoV, I think SARS-CoV has already made a lot of problem with this world, and everyone knows about it. It made the world completely halted, both economically, health-wise, economic-wise, and also the society-wise. Everything was completely divided. So to start with, the highly pathogenic, the CoVs or the coronavirus belongs to the family of Coronaviridae and has been divided into 4 genera: alpha, beta, gamma, and delta. So SARS-CoV is an RNA coronavirus responsible for the coronavirus disease outbreak, and it is proven to be a novel pathogen for the COVID-19. So somewhere here, this coronavirus, somewhere it belongs to here, where it is predecessors, maybe this SARS-CoV-1, SARS-CoV-2, the influenza virus, and the MERS one, and based upon the size and structures, something like this as follows. Some important key important words which I just want to reiterate here, because most of them know, because my presentation deals with most of the things, it will be something like this. Now, what do you mean by mutation? It refers to a single change in the virus genome, happen very frequently, but only sometimes, the change of the characteristics of the virus. What do you mean by a lineage? Lineage is a group of closely related virus with a common ancestor. The SARS-CoV has many lineages. That is the most important thing. Everyone should follow that one. What do you mean by a variant? A variant is a viral genome that may contain one or more mutations. So that's the most important thing, and that is the one which we are dealing with nowadays, because there are a lot of variants now. So a group of variants with similar genetic changes, such as a lineage or a group of lineages, may be designated by the public health institution as a variant of concern or variant of interest. So there is a subtle important differences between what exactly you mean by a lineage, what exactly you mean by a mutation, and what exactly you mean by a variant. Now, what are these important variants which I think I will be talking today? These variants being monitored, it's also called as VBM. This is monitored by the United States, and it will be continued to be monitored, characterized by the federal agencies. And the variants of interest, currently there are no SARS-CoV-2 variants that are designated as variants of interest. Variants of concern, the current VOC in the United States are being closely monitored by the federal agencies, and of course, the variant of high consequence. So what do you mean by this variance of the variant with specific gene markers that have been associated with changes in the receptor binding, and it is also reduced with the reduced neutralization of the antibodies, reduced efficacy of the treatment, and predicted increase in the transmissibility or the disease immunity. So what are exactly its attributes? The specific genetic markers that are predicted to affect the transmission, the diagnostics, or the immune escape. Here comes the most important one, which I think we will be discussing today, the immune escape, where there is a chance that some of the important variants of these viruses may go escape to the vaccination also sometimes. The evidence that is caused by an increased proportion of the cases with unique clusters, and of course the limited prevalence. So what are exactly the possibility or the possible attributes to the variants of the reasons include the impact on the diagnosis and vaccines, widespread interference with the diagnostic test targets, substantially decreased susceptibility to one or more class of therapies, reduced vaccine-induced protection from the severe disease, increased transmissibility, and also the increased disease severity. So coming to this variant of high consequence, it has got a clear evidence that the prevention measures and the medical countermeasures have definitely reduced the effectiveness relative to the previously circulated variants. And that is the reason why we should have to make a note of what exactly are the variants so that we can have a reduced effectiveness related to the previous circulating variants. So these are the important attributes for this high consequences are as on this slide, please. And these are the ones which are being monitored by the United States of America, by all the things. So another important problem this SARS-CoV-2 created in the whole world was two important universities started practicing here. One was, of course, the CDC, and another was a WhatsApp university, where I think it made all our population completely confused regarding it. What exactly the COVID? What exactly is the treatment of the virus? What are the mutations, etc., etc. So one of the important statements which are commonly seen in our WhatsApp nowadays is that variants of concern divided into 4. One is alpha, beta, and gamma, and delta. So these are the variants of interest. It was first detected a B.1.526 in New York, Nigeria, Brazil, and this is the one which is seen in India, B.1.617, where there was the number of spike mutation was about 3, and the attributes are something like this. So these are some of the important statements of this variants of interest. To start with, I think I should be telling something about the surface of the virus protein, where exactly the mutations is happening and why exactly the variants are happening, and what are important reasons why there is also a problem with the reinfection, and also there is a problem with the vaccination problem. Vaccination, even though the people are taking vaccination, there is a disease for few. Why this is all happening? So in order to start with some of the important basics of this one, there is what is called as a spike protein, a membrane protein, and an envelope protein, a nucleic capsid protein, and of course that's what the genome is made up of. And the most and the very finely done thing is your spike protein, where majority of the mutations are attributed to. It is divided into S1 and S2. The S2 is the one which is going to give the conformational change and thereby causing entry of this virus inside the cell membrane, whereas the S1 is the one which mainly acts as the receptor binding protein. So this is how this protein has been completely divided, and the whole of this mutation wise and the most of your diagnosis wise is mainly based upon this structure, which is mainly composed of S1 and S2. See, what are the important things? It consists of RBD, the CTD, and NTDs, etc. Most of your diagnostic things and most of your viral drugs and most of your vaccines are again produced these important proteins. So it contains about 23 open reading frames, and these are the one which is responsible for production of non-structural proteins also. So the genome, it encodes 4 structures, that is spike, membrane, envelope, and nucleocapsid proteins. And this is how the virus enters into the cell. And there is what is called as endolysosomal pathway, where exactly the virus enters into the cytoplasm, and thereby it enters into the nucleus, and thereby production of lot of proteins of these viruses. Now coming to this mutations, most of the mutations that occur in the SARS-CoV genome have no notable effect on the spread and virulence of the virus, and hence on the course of the disease. This is the most important statement which I want to make here, because only very, very few mutations are the one which are responsible for the virulence of the virus. Okay, that most of the people make a havoc of these mutations. It's not so, and that is the common phenomenon which mainly occurs in every single standard RNA virus, including the coronavirus. So the mutation may lead to, the may is the most important one, may lead to the virulence of the virus, could lead to an increase in the severity of infection or failure on the effects of the vaccine currently being employed. This is mainly because the viral signals may escape the immune protection which originates from the preceding infection or through the vaccination. That's how this mutation is mainly occurs. See this one. There are currently about 4,000 mutations which are going on in this S protein gene. There are few mutations in the region called as receptor binding motifs. Okay, and that is the one which is responsible for the viral entry through its interaction with human angiotensin converting enzyme. So if you do some manipulation here, there is a chance that we can curtail the virus entry into this. So about 58% of all the mutations on the RBD potentially increase in the complexity of the antiviral drug and of course the vaccine development. This overall analysis suggests that the mutation in the RBD enhanced the binding of the S protein and CE2, leading to more infections or infectious SARS-CoV-2. So there are some important mutations in the RBD and other domains of the S protein. That's something like D614G. This was first detected in Germany and China in late 2020. The mutation increased transduction in many cell types, including the lung, liver, colon cell. It's more resistant to the proteolytic cleavage. So it is about 4 to 9 times more contagious. And this is the one which has become a lot of havoc in this part of the world. This is another type of mutation which first occurred in the S protein in the Belgium. S943 mutation is a result of the combination of different virus in an infected host. This is the one very, very important thing which was observed in the Belgium region. There was another important mutation called V483 mutation, which was first seen in the North America. And the RNA application site was resulting in the mutant site causes virus to mutate in the host, resulting in strong drug resistance. See, this is how this statement made something called as mutation may be responsible for a drug resistant, and this was the first important mutation whereby they saw that there is some sort of drug resistant, especially due to this mutation. E484K mutation observed in South Africa. It is a rapid spread mutation found in the variants of South Africa and Brazil. And the mutation in the S protein suggests that the virus is further developing and may become resistant to vaccine. This is another important mutation wise. This paved the way that yes, there is a chance of what is called as resistant to vaccine for this important virus. Another mutation located in the RBD, particularly concerned with the 2 reasons: it increases the affinity to the ACE2 and an impact association of the receptor binding neutralizing antibodies. And this important mutation also gave away a clue that there is a chance that this mutants may escape your daily diagnostic techniques of detection of this SARS-CoV. L452 mutant first detected in Denmark. See this one. This mutation weakened the antibody neutralization and increased the virus ability to infect. So this was another important phenomenon which was noted by this L452R mutation. And there was another mutation, Q677 mutation, noticed in New Mexico and Louisiana. This mutation was enabled SARS-CoV-2 enter into the human cells more easily due to its Q location. So this also paved the way that this virus is going to look around lot of windows, especially when entering into the AC cell or AC through cell. This potentially causes a breakdown of disulfide bridges in and around this RBD. It is thought to be associated with increased infection or spread. Of course, yet the studies are still going on with this type of mutation, P681. And some SARS-CoV-2 variants recently associated with the rapid spread, which is causing a lot of things, especially in Russia and England. The B.1.1.7 variant was first seen in UK. A short time it was also seen in India, Netherlands, Bangladesh, Turkey, etc., etc. So this was a very important one because it started proliferating around the world very, very fast, and you would have seen all this variant in a very fast moment of time. Variant has been detected in the US and more than 30 countries, predominantly in England. B.1.1.7 variant exhibits rapid growth and transmission, has the potential to affect the healthcare, pandemic management, and prevention. So what happened is suddenly the cases started increasing. We were not having enough lab to detect it. We were not having enough the hospital management to contain the pandemic. That's what happened somewhere around, especially in the second wave, somewhere around December or January in England. Now, the reason for this rapid spread, they attributed to N501Y mutation, increasing the receptor binding affinity. The variant also has a deletion at the position 6970 of the S protein. So finally, the variant appears 30 times higher mortality rate along with the other variants of the SARS-CoV. So this was another important thing which they derived from especially the England one, where there is a chance of 30% higher mortality as per the other COVID SARS-CoV-2 was considered. So this is another important one, originated in South Africa, and this started to impair the efficacy of the monoclonal antibody therapies and vaccination, thereby giving a clue that just preparing one set of vaccine for a particular antibody may not be enough for curtailing this COVID-19 spread. This is mainly because of many mutations reside in the antigenic supersite of NTD1617 or in the ACE2 binding site, which is a major target for the potent neutralizing antibodies. So this is another important one which was detected in the Brazil, and this is the one which was detected in Denmark, and this is the one which was detected in New York, and this is the one which detected in California. So if you take these type of mutation, characterizing of the genetic variants of the SARS-CoV is very, very, very crucial for tracking and evaluating its spread across the countries. Because if you know certain important variant is there by one country, it is very, very easy to tackle or curtail its spread. The genomic ability of the SARS-CoV-2 samples scattered around the world may be under geographically specific etiological influences. So the continuous monitoring of the mutation is very, very important in tracking and the movement of this variation. So this was another important thing which was observed in the North America. And yeah, this was the two important clinical isolates from India was sequenced. The sequence analysis was performed on S protein of the Indian isolates according to the Chinese Wuhan isolates. The point mutations were identified in the Indian isolates. One of the two isolates were found to harbor the mutation in the RBM at position 401. This is a very, very important documented one which I am presenting today. So what the future holds, since SARS-CoV virus is first emerged, drug trials and vaccine trials has to be continued. See, try multiple therapeutic combinations in different mutation type and to compare such studies, preventing possible pathways before the virus mutates. That's the most important one, which the future holds for this disease. And it's very important to curtail the spread and also the pathogenesis and the infectivity of this virus. So finally, a broad spectrum inhibitor has to be developed in a conservative target is a viable method for developing anti-COVID therapeutics given the high rates of mutation and recombination observed. So finally, some important things which I think we have to remember now is the S glycoprotein is the RBD is the critical determinant of viral tropism and infectivity. So most of the vaccines or the diagnostic technique, it completely revolves around this S glycoprotein RBD. Mutations in this region will change the affinity of the RBD and show the different infective consequences of this strain. Mutations, which are of the most largest obstacles in the development of antiviral drug and the vaccine formulation, have, of course, a crucial role in preparation, administration, and follow-up of vaccines and antiviral drugs. And finally, the mutation rate derives the viral evolution, that is the one which clearly happens to the virus. So clearly, indistinguishable mutations of the viral genomes have emerged in the different geographies. So most of it is supported by the clinical findings. And my final thing is to modulate the virus pathogenicity, potential drugs targeting that site can be designed depending on the localization of a given mutation. So in order to prepare a vaccine or in order to prepare a drug or in order to curtail what exactly is the infectivity or the pathogenicity of the disease, the mutational agent of the virus knowing is the most important one, especially in order to mediate this the pandemic proposal of this SARS-CoV. That's the most important one which has to be appreciated. I think thank you very much. I think I took 20 minutes, 25 minutes.
Yeah,
I rushed a lot, I know, but yeah, the time given was very less to me. But no problem. I think I made this one. The structural protein or the spike protein is the one which is a very, very key factor for the diagnosis of SARS and also prevention and also the treatment. Thank you very much.
Yeah, it was a wonderful presentation, Professor Jaisima. Very crisp, though its time is short, but then you could make it. It's a wonderful presentations. And what I could understand is it is not the mutation alone. Mutation, 90-95% mutations go unnoticed. So on those who are responsible for creating the problem should be monitored in localized ways. So for example, in India, which are the kind of mutations we are having, or in Australia, or so on and so forth. So that's a wonderful thing. And shall I ask Professor Kiran to talk about emerging trends in vaccinations, possibilities of vaccine resistance, current strategy, and the futuristic model. Ma'am, you can speak as such also. No need of presentation. If you don't have any presentation, I mean, not required at all. We are just sitting here to discuss some of the issues, and then after your discussion, I'll be asking you a few questions and then question our sessions, and we'll open to the public. So you do no need of having any PPT. Yeah, please.
Thank you, Dr. Biswajit, for introducing me to the audience. And I really congratulate Dr. Jaisima for giving that nice presentation about that mutations going on in the SARS-CoV-2, the virus responsible for this ongoing pandemic, COVID-19. Now that I will just continue from there only. Now we have seen that what Dr. Jaisima has told us, that different mutations are going on, and currently there are so many variants circulating. So in our country, India, here, so we are seeing many of those variants, but the predominant one, the Delta variant, is there. And throughout the world, we are managing sick patients, though with that therapeutic modalities. So, but as a preventive measure, so throughout the world, we have the different vaccines available to target against this virus. Now, the vaccines may be in any form. The vaccines, they may be the viral vector vaccine, they may be DNA vaccine, mRNA vaccine, which are there now, the Moderna and Pfizer. Subunit vaccine is there. Clear? So all the vaccines, what do? So they, as my co-speaker has just now told, that the virus, the main function of the virus is when it enters, it attaches, the spike protein of that virus is attaching to the receptor binding domain here. So of that ACE2 inhibitor of the host, wherever the ACE2 inhibitors are there. Now the thing is, and our body is mounting the immune response against that. Main immune response, which is important here, is the neutralizing antibodies, so that we can neutralize the virus which is entering our body. Though the cell-mediated immunity, on the other hand, is also contributing for the immunity, but main part is the neutralizing antibodies here. Now, with this, all these vaccines, whatever vaccine we are using, the end function of that is to maintain the neutralizing antibodies, to increase the neutralizing antibodies in the person's body as a preventive measure, so that as and when the virus attacks that person, so the body is able to neutralize it and the person doesn't get the infection. So with that aim, we are bringing out so many vaccines. Now, the big question is, because we have already seen so many strains are there, so many variants are already there circulating throughout the world. Now, the big question is, will there be a variant in future for which the vaccine will not be working, or for which will become resistant to the existing vaccine? Then how to tackle that type of variant? So that is the big question of today. Here, so I have gone through the literature, and we all know that mutation, actually all the viruses, they undergo mutations. Mutation, it is not the new thing in any type of virus. Some viruses, they mutate more, like RNA viruses will mutate more. And in the RNA viruses, single strand viruses will mutate more as compared to the double strand RNA viruses. Then in the immunosuppressed individual, so because that there will be continuity of that infection for prolonged time, so there the mutations will be more. Then the other thing is, there is an inverse relationship with the genome size. More is the genome size, less is the mutation. Less is the genome size, more is the mutation. Then there are the host factors also which contribute towards the mutation. The host protein which help during the viral replication. Clear? The host immunity itself, it will contribute in one way or the other. Then there is the intrinsic polymerase activity also of the viruses, which help in the mutation for the virus. Now, why the virus is mutating? Virus is mutating actually for its own survival. So virus will mutate, so because it also needs to survive. Now, the thing is, in this fight of virus and the host, who is going to win? So that is the big question. For that thing, now what do we need? The vaccines are doing their own part. There is a very beautiful study by Simon A. Raleigh from University of Austria. It is published in Scientific Report very currently in October 2021. And beautifully, they have shown the way mathematical modeling. With the mathematical modeling, they have shown how the virus will contribute towards that, whether that this vaccine-resistant strain will emerge or not, and when it will emerge. So they have taken the different scenarios. So in a nutshell, I will tell you. So when the virus will mutate more, so they have shown with the mathematical modeling, so it will mutate now. So when more of the population is vaccinated, and in their study with the modeling, they have shown that when the 60% of the population of that area is vaccinated, and when the transmission rate of the virus is very high. So two things should be there. Clear? So that time the mutation will be more. And in that mutation, so what we need that in that mutation, there may be one mutation. so which can lead to the vaccine resistant strain. So that only the time will tell us. So there is no clear-cut answer to that yes, by this time that vaccine resistant strain will emerge today or tomorrow. We can't tell like that. We can't predict. So, but then with the modeling, they have beautifully shown that these can be the scenarios. So the best scenario for that viral mutation is when 60% of the population is vaccinated and the transmission is very high. Now, when this will happen? So now you see that, let us take that scenario of our country. Now, in our country, India here, so what is happening? So here we have that 60% of the population so is vaccinated, but with that one dose. Double dose, it is 25% to 30% only. So then now, if with the both doses, the vaccine, the person have taken, clear, so they are immune to that, they have that good immunity. So then there is a evolutionary pressure on the virus also. So because it is bringing the virus under too much pressure to survive itself, clear? So virus, what it needs, it also need to survive now. So it will mutate more for its own existence, clear? So that is the concept behind that. Now, the other thing that they have told, the transmissibility should be very high. Now we have seen the various mutations, which that Dr. Jayasima just now had told. Delta variant, let us take, which is now spread throughout the world. Delta variant, it is already proved by various studies, by various researchers, that it is about many-fold, it has increased transmissibility. Clear? So now, if that 60% or much of the population is vaccinated, virus is already under pressure to exist. It will mutate, okay? It will mutate for its own existence. And then a strain comes out, which is highly transmissible. Then even if when few people are unvaccinated or they have not taken the vaccination full doses, those are the people, those where the virus can go and replicate. And once it replicates, with each cycle of the replication, there is a chance of mutation, because we know that in the DNA viruses, there is the chance of mutation is the 10⁻⁻8 to 10⁻⁻6. Clear? Substitution per this nucleotide substitution per cell infection. Clear? So, and in the case of RNA viruses, it is even higher. It is 10⁻⁻6 to 10⁻⁻4. So in that scenario, it is very, very important. So what we can do in this current scenario now when the vaccination is going more, so what we have to do now? So our thing should be to take care of the non-pharmaceutical things, like hand washing is there, like the masking is there. We have to keep on putting our mask. So we have to keep care of our hygienic measures like hand washing. And then we have to keep the social distancing. Clear? So these three measures, these are called non-pharmaceutical measures for the prevention. So these have to be taken care, keeping in view the current scenario. When the vaccination is going more, and the virus is under more pressure to mutate, clear? And so already the more transmissible strains are circulating. So this is every person duty in the current scenario to take care of those non-pharmaceutical measures to cut down the rate of the mutation, even if it comes, because mutation, even if it comes, there are high chances if we don't give that what you can say, that culture site or the replication site to that virus. So if it doesn't replicate, if we take care of this social distancing and we keep the mask on, so we are not giving the opportunity to virus to attack the other person. So the mutated virus, which has already come after infecting one person, it will not get transmitted to the other. Clear? So less transmission will be there, and with less transmission, the mutated virus will die of its own death. Clear? So many times it is happening in many viruses, they mutate, and many type of mutations they die off. Clear? So they go off after some time. So that should be our first duty in the current scenario, keeping in view. So those things are very important. Now, what other things we can do? One, I have told you that non-pharmaceutical intervention should be always up. Clear? So other thing what we have to do, we have to concentrate on more and more vaccination. We have to spread the awareness among the public. So wherever we go, whomsoever we are meeting, so no matter whether they are scientific community or we meet the other person also, go on spreading the awareness. Each citizen of any country has to contribute equally. Then only we can take care of this. We can cut down the rate of this pandemic. Then other thing is the rigorous genetic sequencing has to go on. Why? Because they can keep a track of the emerging strains. So which type of strains are coming up, how they will behave. So this is very important. Already the initiatives are taken up. They are taken up at the global level also. It is done with the global, GSAID is there, that global initiative for this sharing all influenza data. So that is there. And country-wise also that these institutions have come up, the consortiums have come up. Clear? So the sequencing has to go on at a very extensive manner. So then there should be a global coordination to bring this, to cut down the level of this mutation. Without that global coordination, now we must remember, so even if one country is fully vaccinated, whereas the other one is not, clear, so they are not able to get that vaccine, or their population is less vaccinated, every one of us is at the equal chance of getting that, again, the infection. Pandemic will not go. Clear? For the pandemic to go, each country, each individual has to get vaccinated. So then only we can cut down the pandemic. That should be our aim, and that all that global coordination is needed for that. So then the other thing is, so with all these measures, I will say, so we can bring down that current pandemic now. So these were about the current scenario, that how it is going on. And now I will take up the question then later on. So these were the few important points that which I want to highlight to cut down the pandemic rate.
Okay, thank you, ma'am.
Thank you.
Now I'll have a few questions for sir, and then I'll come back to you. You can take some time. So my question to Professor Jayasima. Sir, today's newspaper, you might have seen Times of India, and that there is a news about the booster dose. After second dose, people can go for booster 6 months after the booster, 6 months after the second dose. Now your comment on that.
As Kiran Chawla madam told, I think every person has to be immunized. That is the most fundamental thing that has to happen. Second one is, especially for the people in the healthcare workers, there is a bit of confusion, or there is a bit of pressure also to go for a booster dose, especially those people who are working in the healthcare aspects. I think definitely, probably, I think they are seeing more number of cases, more number of transmission, more number of sample collection thing. Most of it is happening in the hospital. I think it is going to come. Probably the booster dose may be effective in the course, probably.
Okay. We have some inflammatory markers during the COVID infection process. Say, for example, C-reactive protein, D-dimer, IL-6, ferritin, etc., etc. Now, do we have a risk score analysis that how to score them, and then the severity of the disease? Can we come out of that? Say, for example, in HRCT, they have a CORAD score. Now, do we have a score on inflammatory markers?
No, actually they have started developing, sir. They have started developing this, especially after that CRP, which is one of the most important one. The second and the most important thing, what they almost related the severity of the disease was with the procalcitonin. That's another one, a very important marker which they started. I think it has started one. Probably, I think in the due course of time, all will be completely tabulated properly, and it will be coming. It's on course. It will be coming.
Yeah. So next question will be, what can be the diagnostic period to detect SARS-CoV-2 infections? The shortest period of?
See, that actually depends upon the virus course and the technique, what exactly you have for the diagnosis. Right now, I think in my experience in our lab, we have almost detected from day 2 to day 3 up to day 12, especially for the PCR one, especially for the detection of this the genes, the ORF genes and the RDRP gene. That is the one which you are following now, this one. And the antigen, yes, especially for the antigen aspect, we are detecting from day 6 to probably the next. 6 is the date.
Okay.
We cannot detect before 6th date.
Okay, that's the gene of interest. Now, in such situations in the market, we have 2 gene, 3 gene kits available, right? Now,
Right, sir.
which is the better ones? You say ORF, N gene, E. I mean, which is the better of either to go for 2 gene or 3 gene kits? I mean, apart from internal control.
Yeah. I think in the market there are about 2 genes are also there and 3 genes are also there. I think we have worked both on 2 genes and 3 genes, but especially for the microbiologist point of view, I think 3 gene, I think it's a bit of little headache for us to diagnose. But when it comes to the diagnosis proper, yes, I think they say the 3 gene one is more specific and sensitive compared to the other 2 genes.
Right, sir. Now, this currently available RT-PCR kits, can they pick up the SARS-CoV variants, different variants?
No, I think till now what the kits what I have used, I think it's not picking any variants.
Okay.
Only the genes, I think it's picking up.
Right. I mean, ICMR has not given any guidelines.
Yeah. No. I think it has not. I think it's still in pipeline. Still in the pipeline.
Okay. Okay. Shall I ask few questions to ma'am?
Yeah. Yes.
Yes. Sure.
I think she was talking about the sequencing and just some time back. Now, does gene sequencing will play a major role in managing variants in future?
Yes, of course, because this is only the method which can screen the patient sample and can see that which are the variants emerging and what is the in that area, which strain is circulating. So based on that, so the various measures can be taken, and the whole effort is now, the whole thrust is over this sequencing to see that what are the various variants which will be in future also, which will come up, so that the appropriate measures can be taken up in time.
You think this information should pass on to the clinicians or the people who are taking the decisions in treatment protocol?
Yes, I think all scientific community is aware of that, because now so much media is there and it is spread by the media everywhere. So I don't think any scientific person will not know this, but only thing is, because sequencing, it is costly affair. So for patient, we can't do like that. So it is because for diagnostics, as far as diagnostics are concerned, so clinician is concerned just whether the patient is infected or not, and what is that level of sickness. So that if the patient is very sick, so they can take that appropriate measure for that. So, but based on that variant, the treatment will not vary. So that is the as the clinician perspective is concerned.
I came across such a patient. He demanded that my things to be sequenced. So that's a good question. Okay, next is, ma'am, have you come across vaccine-resistant transmissions of SARS-2 variants?
Vaccine, pardon me?
The question is, have you come across vaccine-resistant transmissions of SARS-2 variant?
Till today, there is no vaccine-resistant variant seen till today. So nothing has been reported so far.
That's true. And next question is, does variants have impact on vaccinations? Of course, you have told in your
Yes, actually, some studies have been done. So I have went through those studies. They have seen that different variants, they have constituted those variants actually on that different virus, the genetically modified viruses. So, and they have seen that these variants, they have the impact on transmissibility, their virulence also. So they have seen that. With the various by the animal model studies, they have seen the effect on hamsters also. They have done with that this neutralization testing also. So I have gone through the study. So yes, they have the effect, the various variants. Just now we know that's why Delta variant has come very important, because it is proved now that it has many times more transmissibility as compared to that native, the first virus which was detected, the wild virus. Now in future, we can't predict what type of variants will emerge, but yes, it can happen that, and possibilities are there. So that's why, so more thrust is on that, getting the people vaccinated, and thumbs up for that non-pharmaceutical interventions to curtail the spread of the infection and for emergence of these variants.
My last question.
Yes.
Emergence of vaccine-resistant strains may come up too early. That's what the news agency says. It can come any day, any weeks, any month.
So that's what I'm telling, that you can't predict the time. No one can predict the time. It is only the possibilities are kept. And that and the paper I already mentioned by Simon from University of Austria, they also very beautifully, with the predictive modeling only. So they have given the prediction that yes, it can emerge, but no one can predict that what will happen tomorrow. So that is.
Sir, one more question to Professor Jayasimha. Just one question about antibody you just talked about some time back. The currently available antibody kits that is available, can it pick up the variants?
No, sir, no, it cannot. No, yeah. it will not. See, the problem is, as Madam told, the routine PCR itself nowadays, I think we are having some problems with it. The next thing is going to be the sequencing. That is the one. Even though the cost is the constraint, that is the way to go about for the curtailment of the pandemic. We have to sequence the cases, all cases, or at least that cases which is completely surrounding your area, and then you have to formulate the policies, and then of course you have to treat and also take measures to curtail the infection.
That's true. But the ICMR never allows people to go for, the lab to go for sequencing, because they kept it for themselves, or the limited labs. So that thing is not allowed right now. Am I correct?
Right, sir. Right.
The sequencing is being done, but at the central level. So they the consortium is there. So they are doing, and that 5% of the positives they are getting. So, but it need to be increased, the sequencing.
So that it will be more broad-based and more information will flow in. So now it's open to the audience. If they have any questions, both our panelists are there. So you have a chat. I think there are 4 questions are in the chat box, and just have a look on that. Professor Jayasimhan, can you answer this? The last one. You are muted, I think.
since vaccine-resistant strains emerge under selection of pressure of protective antibodies, how about revaccination with a different vaccine or any other proposed strategies for the control of these mutants? A wonderful question asked by Dr. Yogesh Babu sir. Yes, for this, I think we have to go to the next level. Next level of diagnosis is the key there. What exactly has to be done and how to recognize the mutants and all, and what exactly, I think a lot of vaccines has to be manufactured. More number of vaccines should come, and more number of vaccines to target a particular protein has also to be devised. I think we are getting vaccine on mRNA, we're getting on one protein, two protein like that. There should be a system of a cocktail of what is that one product which is going to act on the proteins of the virus. That is the answer for this.
Yeah,
Adding, just 1 minute. Adding to you, Dr. Jayasima's this thing only. Yes, that already research is going on. The people have started seeing that more vaccines against the different strains also which are emerging. So against 1 or 2 strains already it has been started. But now keeping the current scenario in mind, so the thrust should be the full vaccination to the full public. Because we must remember, no one is safe until everyone is safe. So there will be probability if we, the people who can get the vaccination can acquire the vaccination, they are even after getting the booster dose. But at the same time, in other countries, the people are not vaccinated. So that is a futile exercise then. So then everyone again will be at the same level. So you can get the infection again. So because you are giving the chance, unless until everyone is vaccinated. So then, and you are taking care of that proper other measures, preventive measures. So you can't curtail this pandemic. You can't bring down the infection rate. So currently, so that has to be kept in mind. But yes, research is going on for those things.
Sir, one question, that booster question has come. You can see that. Jasmine sir.
Yes, sir.
Same question has come. If you want, you can respond.
I think as Madam told, I think each and every population has to be immunized, more than 60 to 70%. And the second one is the importance of non-pharmacological strategies, what Madam has told. If we keep that in mind, probably there may not be any booster dose required. Who knows?
Ma'am, one more question. Are vaccines available in India approved by WHO?
Yes, now that we have the Covishield and Covaxin, both are approved by WHO. So all are approved now.
Emergency, yeah.
Yes, emergency.
Yes, sir. Sir, one more question that is asked by Rupian. Can you respond, ma'am? Mr. Puneet? After taking the second dose of vaccine, also some people are having COVID positive. Why?
Yeah. So Dr. Jasima is replying to
that.
Okay, for me. Okay. So actually that vaccines which are available in India, I think you are from India only, Mr. Ronit. So then the vaccines in India, they are not 100% protective. So already if you go to the efficacy of that vaccine, so depending on the vaccine, the efficacy vary. But these vaccines, they protect us against the severe infection. So it is not a guarantee that you will not get the infection again. Already there are reports for breakthrough infections. So these are given the name as breakthrough infections. So there are reports. So, but only thing is the government itself is telling that these vaccines, they protect against the severe infections only. So there is no guarantee that you will not get infected again.
That's the best answer you can give. It will protect you from the severity. Yeah. So that's question again come today. I read that we can use booster after six months. The same question has come. It has been answered twice. So if any other questions, and if not, then the organizers to respond. Hello. Yeah, Khushbu.
Yes, sir. I guess there are no more questions now. Most of the questions have been answered.
Yeah, very well answered by
Yes,
both
sir,
our
very well
panelists.
answered. Yeah.
Excellent.
Yeah. So, sir, if you allow, we can wind up the session.
Oh, yeah. So thank you very much, Professor
Thank you very
Jaiswal.
much, ma'am. Thank you very much.
I personally thank all the people and all the organizers for having us here and giving us the opportunity for this nice session.
Yeah,
Thank
Thank
you.
you all.
On
Yeah,
behalf
thank you
of
all
Cipla
organizations,
and
Biswajit.
Cipla.
Yeah, thank
Thank
you all
you, sir.
organizations, Vishwajit sir, and great to share as a co-speaker with Kiran Chawla madam. Namaste.
Yeah.
Thank you.
Oh,
So on behalf of Cipla and Cipla Diagnostics, I would like to thank all the faculty members, Dr. Biswajit, Dr. Jaisima, and Dr. Kiran Chawla madam, for the very excellent and very knowledgeable session. And lastly, I would like to thank all the attending doctors for sparing their time and attending the session. Thank you.
thank you.
all.
Thank you all. Bye.
Thank you.
Bye. Have a good